Hi, my name is Todd Nelson, and am a Fraser Health Research Assistant, and I will be presenting our paper on behalf of the MRI on ME research team. So our paper is titled Brainstem Abnormalities and Myalgic Encephalomyelitis, Chronic Fatigue Syndrome, a scoping review and evaluation of magnetic resonance imaging findings. So first, I'd like to acknowledge our research team. We have clinicians, scientists, and patient partners from Fraser Health, BC Women's Hospital. Research patient partners are from ME/FM Society of BC. So the authors of our paper include Dr. Zhao Song, my supervisor, myself, Nancy Zhang, and Hui Guo, and Louis Nicole. And we'd like to acknowledge our funders, the BC SPORE, Surrey Hospitals Foundation, NSERC, and BioTalent Canada. So in this presentation today, the outline will be us discussing the background, objectives, methods, results, conclusion, and dissemination plans. So first, our background on ME/CFS, or myalgic encephalomyelitis chronic fatigue syndrome. It is a multi-system condition that affects the central nervous, cardiovascular, and immune systems. And over 17 million people worldwide live with ME/CFS, and of those, around 580,000 are Canadians. Several common symptoms include post-exertional malaise, which is just a worsening of symptoms and fatigue post-exertion, whether that be physical, mental, or emotional. Autonomic dysfunction, which includes things like orthostatic intolerance or postural tachycardia issues, cognitive problems such as attention and working memory impairments, and a substantial reduction in activity engagement. And then we move on to brainstem. The brainstem is a compact structure between the cerebrum and the cervical spinal cord, and it's best known as the hub relaying information between the brain and the body. It is comprised of 3 areas, generally the midbrain, pons, and medulla, and consists of both gray matter, which is generally cell nuclei, and white matter, which is the axons of those neurons. So there are several brainstem dysfunctions that could be associated with ME/CFS that we first recognize. Several examples could be impaired brainstem reticular formation, which an impairment there could lead to impairment in sleep and consciousness, and thus impaired cognitive function. Another example could be a damaged vagus nerve, which the nerve innervates the brainstem, which means it has connections with the brainstem, and that could lead to autonomic dysfunction. We also have impaired dopaminergic brainstem areas, which could then lead to also impaired motivation, concentration, and learning. And finally, perhaps an impaired medulla and pons could lead to cardiac and respiratory symptoms, as those are the centers that really control those types of things. And moving on to MRI, also known as magnetic resonance imaging. What it is that it uses a strong magnetic field to create imaging contrast between different types of tissue, whether that be white matter, gray matter, or cerebrospinal fluid. And there are several types of MRI. There are structural MRI techniques, such as T1 weighted, T2 weighted imaging, DTI, and these are really used to reveal the brain anatomy, lesions, and structural changes. We also have functional MRI, also known as fMRI, used to really study the functional connectivity of brain regions and between brain regions, and the brain's response to stimuli. We also have magnetic resonance spectroscopy, MRSI, which is really used to reveal information about brain metabolites and brain tissue temperature. Some of the key things that really are of benefit of MRI is its non-invasiveness and its high resolution. And because of those reasons, it really makes MRI preferable for clinical diagnosis and research. And because of all these different types of MRI techniques and its benefits, it's been used in the past to really understand ME/CFS. Moving on to our study rationale. So currently there are no FDA or Health Canada approved treatments or cures. And this is presumably owing to the unknown condition's pathology and mechanisms. And we're starting to see emerging reports of the brainstem involvement. Some things include reductions in gray and white matter volumes in the brain, but with inconsistencies regarding the brainstem structural changes. Reports have also shown increased brain region recruitment and activity when dealing with those symptoms, but again, with inconsistencies. We see reports showing changes in brain metabolites, bold signal complexity, and functional connectivity. And as I mentioned before, there is a potential associations between the brainstem dysfunction and ME/CFS symptoms. And because of these reasons, there really was a pressing need for review and evaluation of MRI findings, really concerning the brainstem's involvement in the condition. So that led to several objectives of our study. And one was to integrate relevant MRI findings of the research field and then to derive synthesized evaluation of the brainstem's role in the condition based on the findings. And we intend to provide new insights to advance the research line and also to inform future solutions for better management and care. And we're seeing that in the literature, it's been suggested that ME/CFS is a long-term consequence of COVID-19. So this is also one of the motivations of the study. So first, methods is eligibility criteria. And what we really looked at were for papers that investigated the brainstem using MRI, and that was original research that had full text, was conducted and published in English, and had human participants. And this is the figure that you see on the right is figure 1 of our study. So now we move on to our search term. So this literature review was conducted by 2 researchers and was done using the Medline database. And we have 3 keyword sets that we initially used. Set 1 was to look for papers that focused on ME/CFS. Set 2 was to look at studies that utilized MRI techniques. And the last one was to look for studies that investigated the brain. And then came the selection of the sources that we would use within our review paper. So we used database filters to filter out things, papers that didn't focus or didn't use human participants, weren't published in English, weren't full text. And we did another database filter to really screen out non-original research. We then moved and conducted a title and abstract read to further ensure that no non-original research was still in our subset. And we also removed articles that were not brain MRI studies on the condition. So from there, that led to a smaller subset in which we screened and evaluated the entire paper to remove papers that did not include the brainstem or its components. And that left us with a final subset of 11 articles. So now, synthesizing our results. So what we did was the findings of each study was grouped into either structural or functional brain changes. And we reported for each study several things, including participant characteristics, MRI techniques, study design, and publication year. And finally, the main findings concerning both brainstem and non-brainstem specific. So this is just the initial results that we found from looking at those papers. And it seemed like MRI was really used to study the condition for decades. But we see the first use of fMRI, functional MRI, in 2018. The majority of the studies included both sexes/genders, and the age ranged from the early 30s to the late 40s. All studies used T1 weighted imaging, which is the structural imaging, while 6 used T2 weighted imaging, and 1 used T2 FLAIR to better understand the pathological changes. 1 used DTI, and all but 1 performed a one-time scan. In other words, they only scanned participants once. So here we see the publication trend. So if you look at the graph or the figure on the right, we see that really the MRI has been used to study the condition for quite a long time, since 1993. But we see from 1993 to about 2010, how all of those weren't focusing on the brainstem. And the first study really investigating the brainstem was in 2011, and that was a structural paper. And from then we see more brainstem focus. And in 2018, we see the first functional paper done with regards to the brainstem. And we see now that in the last year that we looked at, 2021, how all of the studies are focusing on the brainstem. So now the main results of those papers. So the first is structural. So what we saw from those papers were that there was white matter volume impairments in the brainstem and other regions. And just to reiterate once again, white matter is really the connections between neurons. We also saw myelination impairments in the brainstem and other brain regions. And myelination is really what allows for quick access and quick communication between brain regions and neurons. We also saw cortical compensatory brain changes and opposite correlation patterns between MRI measures of brain structures and physiological measures in patients compared to healthy controls. So now moving on to functional results, we saw abnormalities in many different systems. We saw some in the reticular activating system of the brainstem, the default mode network, salience network. We saw abnormalities from the brainstem to the prefrontal lobe and abnormalities between the brainstem and other brain regions that are thought to be responsible for managing fatigue. So in this case, for patients, we didn't see management of fatigue done very well in these connections. So now moving on to clinical assessments. So the majority of the brain changes that were reported were both associated and supported with clinical assessments, namely vital signs, sleep parameters, disease duration, and severity levels. So now, really looking at the brainstem's function, so the functions of the different parts of the brainstem and the condition symptoms and the connections between them. So looking at figure 3 of our study, we see that there's the midbrain, the pons, and the medulla making up the brainstem, and how there are several structures within the brainstem. So the first one in the pink rectangle would be the substantia nigra / ventral tegmental area. And the function of this area is dopaminergic control of motivation, concentration, and learning. So some of the MRI findings that we saw in these studies were one, reduced white matter volume in the midbrain, so where that is, and abnormal axon caliber or axon myelin density in the midbrain. And some of the symptoms that could be associated with this include concentration deficits and fatigue. So next, moving on to the blue one, reticular activating system nuclei. So the function of this area is the control of sleep wakefulness cycles and consciousness. So some of the MRI findings that show perhaps an impairment here is reduced white matter volume in both the midbrain and the pons. We saw functional connectivity deficits between the cuneiform nucleus and medulla, and between the brainstem and hippocampus, and intralaminar thalamus, which is all part of this network. We also saw, again, abnormal axon caliber and axon myelin density in the midbrain and pons. And this could lead to symptoms such as fatigue, sleep dysfunction, deficits with problem solving, sensory perception, attention, and memory. Moving on to the yellow box is autonomic center nuclei. And what this really does is it regulates heart rate, blood pressure, and breathing. Some of the MRI findings include reduced white matter in the pons, varied correlation patterns between autonomic center nuclei, such as the myelination of it, volume, and physiological measures, such as the heart rate, blood pressure, and ME patients. And because this really controls cardiac and respiratory function, we see that possible symptoms may include cardiac dysfunction, such as orthostatic intolerance, postural orthostatic tachycardia syndrome, and respiratory dysfunction, such as abnormal breathing, and finally sweating episodes. So now moving on to the last gray rectangle, and that is nerve conduction. So as I mentioned previously, the brainstem really is in that central hub that relays information from the brain to the body and from the body to the brain. So some of the MRI findings that perhaps suggest impairment here is the reduced white matter once again in the pons and medulla, different brainstem myelination or iron, and connectivity deficits within the brainstem. And this could lead to things like sensory input and motor output dysfunction. So that would lead to things like temperature hypersensitivity, which is seen, slowed motor conduction, and just pain from the body. So future studies. So from looking at the studies that we investigated, we have several suggestions for future studies. And that includes a larger sample size to allow adequate statistical power to detect differences between healthy controls and patients. We'd also like to see basic demographic information of the sample for better data interpretation and evaluation of generalizability, because we know that ME/CFS is quite a heterogeneous condition that affects different people differently. We'd also like to see the use of more and different MRI techniques such as DTI and MRS. We'd also like to see longitudinal studies to better understand the deficits and consequences and the process of the condition. We'd like to see clinical trials examine the effect of standard care and novel interventions. And finally, we want to see studies targeting the role of the brainstem. Some of our limitations include only applying the Medline database. However, it is quite a comprehensive database. We only included reports published in English as that was the author's preferred languages. And we only included reports that targeted the brainstem using MRI techniques. So we didn't look at ones using EEG or PET because that was just outside the scope of our scoping review paper. And finally, to conclude, from the studies we've looked at, data show that there are indeed structural and functional deficits of the brainstem, and those are linked to brain changes and clinical expression. We suggested a model summarizing the brainstem's role and connection with other brain areas. The paper draws attention for increased attention to the brainstem and calls for future research to consider multimodal MRI, improved experimental design, increased sample size, and increased follow-up duration. Some of the implications of our paper include uncovering ME/CFS's pathomechanism and improving management and patient care. Finally, our dissemination activities and plan. So the MRI on ME study protocol, project initiation, and design have been reported at last year's SPORE event and also been reported through the ME/FM Society BC. Our patient partners have prepared media stories to increase the awareness of this publication among the patient and the public. Key findings on the critical role of the brainstem in this condition are raising increased attention in the research field and supporting new study developments and research funding applications. There's current ongoing effort being made with SFU Image Tech Lab to resume MRI patient scans as soon as the lab is reopened. And we aim to produce additional publications to report the results from the patient scans of that project at scientific conferences and journal publications. And with that, I thank you for taking the time to listen to me today.